This is a recording for Biology 1407, Majors 2, the circulatory system, part of Unit 3. All right, so circulatory systems come in a lot of different levels of sophistication, kind of commensurate with the level of complexity and complexity of the animal in question. So we've got a few different varieties that we can talk about. So as it says right here, sponges, cnidarians, nematodes, and all of those sorts of things don't have a circulatory system. They've got relatively simple, thin, high surface area bodies, and in the case of sponges, no real body, that takes care of their exchange needs fairly effectively. As it says right here, sponges circulate water through pores in their structure. We'll take a look at a picture of that in a minute. Hydras use their gastrovascular cavity as their circulatory system in the sense that it is extremely extensive and sort of penetrates all the distal portions of the body. And same thing for nematodes. All right. But when we get to larger, more complex animals that either are larger or that maintain a much higher metabolic activity. So this is a example of a cross-section of a annelid, an earthworm, right here. And that's a substantially larger animal than most nematodes. The biggest nematodes are going to be parasites where food is already digested for them. And you know, they don't need a huge circulatory system, to transmit materials because they live in a resource-rich environment. And this, of course, is a bee right here, which maintains a very high metabolic activity, so it needs rapid delivery of nutrients to its muscles, particularly its flat muscles. So in both cases, these are invertebrates that have a circulatory system that takes care of metabolic needs. In the case of annelids, it's actually closed, so it has arteries, veins, capillaries, and veins in a loop connected to the heart. Whereas in the case of insects, like all arthropods, it's an open circulatory system, a series of arteries leading off to distal regions of the body, and then the circulatory system permeates the tissues of the body. You all should already know a little bit about open and closed circulatory systems. We'll get a little more detail in as we go along here. All right, so here's an example of that sponge right there, and fluid. This is their means of feeding, which also really takes care of their circulatory need at the same time. Water comes in through afferent pores and exit through an efferent pore right here. And there are little flagella-equipped cells all in these compartments right here that pump water so that there's a continual pressure pushing water through the system. Here's the single opening into the gastrovascular cavity of the jellyfish here, like all of the cnidarians, and that cavity can extend pretty significantly, through the extremes of the body of the animal right here. So that takes care of circulatory mechanisms at the same time. Every time this thing compresses the musculature of its bell right here, that pumps fluid around, squishes fluid back and forth inside this cavity right here, which takes care of circulatory needs. All right. What we'll spend most of the time talking about in this chapter right here is vertebrate circulatory systems, and we'll take a look at some evolutionary trends in vertebrate circulatory systems as well here. So we've got 4 different models right here: a fish, an amphibian, a reptile, and a mammal right here. And to that, obviously, I should point out to you that this follows kind of an evolutionary trajectory. In other words, this circulatory system right here is descended by mutation, innovation, adaptation, accumulation of those adaptations to create this relatively complex circulatory system from a simpler precursor here. All right, so this precursor circulatory system, the fish circulatory system, the key features in here, let's see if we can trace this accurately. We've got one large vein, which is not unusual, one large vein returning to the heart, The heart consists of an atrium and a ventricle, so it has two chambers, an intake chamber, the atrium, an output, a heavily muscularized pumping chamber, the ventricle. And then that artery coming out of the ventricle splits to go through each gill, left and right, and then carries on from those gills, carrying oxygen-rich, CO2-poor blood, to capillaries of all of the rest of the body, including the digestive system, all of the skeletal musculature, everything, and exchanges gases with those tissues. So the oxygen is lost, CO2 levels go up. That's the meaning of this red versus blue coloring right here. Nutrients will have been picked up from the circulatory system. Waste products would have been picked up from skeletal muscle. All of that pumped back through here, and where we go ahead and go through our exchanges of gases, exchanges of metabolic materials, and so on and so forth. Some key important ideas right here is blood comes out of the heart under high pressure. High, it's a relative term, but under pressure. And so it's passing through these arteries quickly under pressure. Quickly is the key thing here. Large volume in a short amount of time. When we spread that blood out over a capillary bed, it loses pressure. And that can't really be regained. Hydrodynamics won't really allow that to occur. If we applied enough pressure to get this blood back under high pressure right here, we'd be bursting all of these capillaries right here, so it just won't happen. So the blood moves much more slowly through this secondary artery right here, or arteries, and passes under very low pressure and very slow speed through the body capillaries of the digestive system, of the liver, of the reproductive organs, of the skeletal musculature, and everything, before returning back to the heart to be pressurized and pumped at speed again. So this is limitations, and the key limitation is that exchanges with body tissues through the body capillaries are not nearly as efficient as exchanges are here in the gills, where the blood enters under high pressure and there's a relatively rapid movement through here. So then when we look at amphibians, we see that there's a partial, there's some modifications that partially correct that set of problems right there, because they are problems. So we have blood coming back from the body right here, coming into the right atrium, pumped into the left atrium, or sorry, into the ventricle. There's a central ventricle right here. And then the blood pumped out of that ventricle is split into two paths. One path leads, just like the fish circulatory system right here, to branching arteries that are under pressure, blue because they're low in oxygen, and they pass through the gills or the lungs, the exchange surfaces, and this includes skin of the mouth, of the body, the lungs themselves. If it's an aquatic stage amphibian, this will be external gills. But regardless, there'll be gas exchange right there. And then that blood is brought back to the heart, joins in a left atrium here, and that's pumped back into that central ventricle. And the oxygenated blood and the nutrient-rich blood coming from the digestive tract are mixed together in this one ventricle right here, and then pumped out in both directions. So some oxygenated blood goes back to the gills or lungs for another round of oxygen pickup. Some nutrient-rich blood, or blood with a moderate level of nutrient concentration, is sent through the digestive tract to pick up more nutrients, but also to deliver to skeletal muscle. So key modification here is that the blood flow through this artery, or these arteries leading to the body capillary beds, is under higher pressure. We move blood more efficiently and more quickly, and distribute it over these capillary beds and return it. So we increase the efficiency of this system. A downside is that this split right here allows nutrients, and half of the load of nutrients and half of the load of the oxygen, to go out on either side right here. So we're taking oxygen back to the lungs, which we don't really want to do. We're taking nutrients back to the digestive system, which we don't really want to do. When we look at reptilian circulatory systems, and particularly those of crocodiles and their relatives, which evolutionarily also implies dinosaurs, birds, and all of those guys, what we see is that the emergence of a prototypical four-chambered heart. So there is a right atrium, a right ventricle, a left atrium, and a left ventricle. So that's a four-chambered heart. But there is a point where these are not completely separated. The level of separation here varies quite a bit. And in some groups, there's something of a valve right here that can be opened or closed to kind of control flow. But what we're setting up right here is a much more directed flow where blood coming back from the body tissues, which is going to be rich in nutrients but poor in oxygen because it's come from the digestive system, it's come from the liver, is going to be entering the right side of the heart and pumped out through the right ventricle and mostly go to the lungs. So this is oxygen poor blood being pumped mostly to the lungs, picking up oxygen, returning to the left side of the heart, and it's still got all the nutrients it picked up from the digestive system and the liver right here. And then that blood coming out of the lungs, oxygenated and CO2 poor now, goes into the left side of the heart through the left atrium. Atrium is a receiving chamber. To the left ventricle, the ventricle is the pump, the real muscular pumping chamber, and exits and splits into paths going off to different tissues of the body again. So this is a four-chambered system right here. Quite a bit more efficient because we have high blood pressure blood going to the systemic capillary beds, a collective term for all of the capillary beds in the body that are not part of gas exchange is systemic. Skeletal muscle, digestive system, reproductive organs, skin, everything else basically. So blood going to the systemic circulation is under pressure, moving quickly, much more efficient exchange at the capillary beds, returns under low pressure, pumped out under high pressure to the gas exchange organs, same efficiencies right here, returned under low pressure to the left side of the heart for being pumped out again. We have four chambers, but not complete separation of those chambers right here. So it's much more efficient, but not as good as it could be. What this does allow is for some switching right here. So they can shut off what's called the pulmonary circuit right here. Pulmonary, pulmos is the Latin for lungs. And what this allows with this sort of incomplete separation right here is this can act like a valve, and we can direct all blood to systemic circulation when the animal is holding its breath or underwater. This is really good for alligators, crocodiles, and all of their relatives. And then when they come up and can breathe again, we kind of close off the right side right here, so it just goes to the lungs. Otherwise, right circulation would join into left circulation right here. It just goes directly through the systemic circulation. So they can kind of switch back and forth. They've got some neat capabilities here, but they lose a little efficiency because of it. More capabilities, you always lose a little bit of efficiency in a specific part. Finally, the circulatory system that we see in mammals and in birds, and us theoretically in dinosaurs, at least some of them, the theropod dinosaurs, the two-legged ones whose birds are descended of. The big difference here is we completely separate the left and the right sides. There's no, during embryonic development, there is a connection, there's a shunt, for the same reason that we've got over here. When you were developing in your mother's womb as an embryo, output from the right side of the heart, that blood was redirected over onto the left side and pumped out through systemic circulation because there was no point pumping blood through your lungs while you were in the womb. That's the way it works for mammals and for birds. But after they are hatched or born, that shunt closes completely, and that's a vestige of this structure right here, but that shunt closes completely, and the right side of the heart is strictly receiving blood from the body and pumping to the lungs. The left side of the heart is receiving blood only from the lungs and pumping only to the body, to systemic circulation. Most efficient. So here we've got an illustration of that fish circulatory system again. The atrium is the receiving chamber, the ventricle is the pumping chamber. High pressure blood going to the gill capillary beds, low pressure blood going to the systemic capillary beds. A lack of efficiency, but it's simple. When we look at amphibians, we have the three-chambered heart right here. So the output from the ventricle goes in both directions, goes both to pulmonary circulation and to systemic circulation. It's just split in half 50-50, and it is a mixture of the nutrients coming back from the digestive system and the oxygen coming back from the lungs. That mixture is in both directions. Not quite as efficient as we would want. Then when we get to that reptilian heart right there, We have a pretty effective separation of the two ventricles with some adjustability right here in this connection between the two. And so the pulmonary circuit, blood from the ventricles, from the right atrium, it goes into the right ventricle, and most of that blood successfully goes off to the lungs, and only a little bit of it bleeds over onto the left side of the heart, or vice versa. And same thing for the systemic circulation. Blood from the lungs into the right atrium, pumped out of the, into the left atrium, pumped out of the left ventricle to systemic circulation. And this opening here is adjustable. And then finally, mammalian or obligate four-chambered circulation is normally, there are birth defects where we have a hole right here, but normally there's a solid separation between the left and the right halves of the circulatory system. And again, the advantages of this system is that we take oxygenated blood from the lungs, from the pulmonary circuit, and exclusively send that oxygenated blood to all other parts of the body. We pick up nutrients, along with many other things, and all of this systemic circuitry right here, hormones released by glands, everything, and then return that via the venous system coming back to the right side of the heart and pump that blood exclusively through the lungs to exchange gases. We'll vent off CO2 picked up during systemic circulation. We'll pick up oxygen instead in pulmonary circulation and then send that back around again. All right, a little bit about the circulatory fluid. So again, in open circulatory systems and most other invertebrates, blood, we don't call it blood, we typically call it hemolymph or some other fluid there. And the big deal about that is that it is mostly acellular. So what we won't have is the red blood cells. That hemolymph will have whatever particular sorts of white blood cells that group of organisms has. Vertebrates have a much more complex set of immune cells than all of the invertebrate groups, but they have some cells as well. But they won't have these red blood cells, and instead the hemoglobin or the hemocyanin or whatever the particular oxygen pigment carrying pigment is will be just dissolved into the blood loosely. Like true blood, it is a connective tissue because there are the molecular bases for forming fibers under certain conditions to plug holes in the circulatory system. That still exists. All right, as it says right here, the functions of circulating blood, and now we're talking about blood, which, as you know, again, is a connective tissue, and it consists of red blood cells primarily, and then a much smaller number of white blood cells. And these pseudocells, they are the fragments of mitosis or cytokinesis, cell division, but without mitosis, where we make a subcellular fragment of an original cell. And the purpose of that blood, of course, is to transport things all through the body, to provide a medium for regulation of certain things, primarily heat, so that the blood can transport thermal energy from one part of the body to another. And then protection, in the sense that it carries these immune cells around all through the body so that they are delivered to all parts of the body where they can act locally to combat infection. Plasma is primarily water with a bunch of stuff dissolved in it. What is dissolved in it? Well, fibers for clotting, blood proteins which maintain a certain level of osmolarity in the blood, so a tendency of the blood to draw water towards it. That's primarily the job of albumin right here. And then these globulins right here, which are immune proteins. That's your antibodies. The alpha and the beta globulins are two major classes of your antibodies. And then everything dissolved in there, which is the stuff that would be dissolved in any tissue in your body. So nutrients, waste products, hormones, as it says right here, all of these different salts, some in larger quantities, some in smaller quantities, but all the stuff dissolved in, the tissues of your body everywhere. When you look at the cytoplasm of a cell, these things are present. When you look at the interstitial fluid around your cells, but outside of the circulatory system, these things are going to be in there. And then finally, the liquid of your blood, these things are going to be in there. But then we add in these things, we add in the cells, etc., etc. A little bit about red blood cells. So these vary a fair amount. In mammals, the erythrocytes almost universally lack nuclei, which means they're not going to live very long and they can't really adjust or adapt to any kind of conditions. So they're sort of alive, but they're not really very alive. They're just kind of running on inertia, if you will. The erythrocytes, the red blood cells. So without a nucleus, they can't produce any new proteins, so they can't do any kind of repair or maintenance. They don't have any mitochondria, so they don't consume oxygen either. And so that makes them a relatively low maintenance, because they don't do any maintenance, and low resource consuming cell. And they're really specialized to do just one thing, and that is to move oxygen. Well, one thing, to move gases. Primarily oxygen, but also CO2 and to a lesser degree a few other gases, some of them inadvertently. And because of this, they only live for about 120 days. Your spleen and to a lesser degree your liver are the two main organs for pulling out dying erythrocytes or malfunctioning erythrocytes, digesting them and recycling the materials. By the way, your liver in its recapturing of the hemoglobin from red blood cells, and your spleen salvages this material as well, but the liver pulls it out. Your liver is going to take some of that hemoglobin composition, the proteins, and use that to produce bile for the digestive system. So that's where the bile comes from recycling the hemoglobin, not the iron part, but the protein portion of hemoglobin, recycling that, the heme group specifically, to make bile. All right, a little bit more about cells in the blood. The white blood cells, WBCs, or leukocytes. And there's a number of other words that you'll see associated with these, basically, depending on what particular feature is being highlighted. But anyway, leukocytes, which are collectively the white blood cells. There's a number of different types, and you don't have to memorize the different ones right here, but I do want to describe them very quickly. So neutrophils are the most abundant by far, and these are your primary defense against bacteria, neutrophils right here. The cells can destroy bacterial cells and destroy themselves at the same time while fighting bacterial infections. Eosinophils, and all of these are granulocytes in the sense that they have these little clusters of granular, protein structures distributed in their cytoplasm that they can, tiny vesicles full of proteins that they can exude under a signal. That's why they're called granulocytes. The eosinophils are much rarer white blood cells that are primarily fighters of parasites, and they consume or destroy or neutralize materials that cause allergic reactions in the blood. So if somebody with a lot of allergies, who's suffering an awful lot of hay fever or who has internal parasites, will typically have an elevated eosinophil count. Just like somebody with an active bacterial infection somewhere, if it's big enough, you can see that in just a normal blood draw because they'll have an excess of these neutrophils. The basophils are another specialty white blood cell having, that is targeting parasites and other chemical triggers in the blood. Then you've got your two agranulocytes, which means they don't have granules in them. The most abundant, and actually the second most abundant white blood cell in your blood under normal circumstances is going to be the lymphocyte. You have T and B lymphocytes, and these are the cells that produce the antibodies that are part of your adaptive immune system. And those things target bacteria. They target things that we're allergic to, and allergies are a malfunction to that that immunity right here. And importantly, they target viruses and cells that are infected with viruses. And then last but not least by any means are the monocytes. In your blood, these cells are referred to as monocytes. These cells leave your blood and aggressively patrol tissues throughout your body, and at that point, they're called amoebocytes because they move on their own like an amoeba, like those protistan single-celled animal-like cells that have no particular shape and can move across the surface. Well, that's what these guys do in your body tissues. Neutrophils also move out into your body tissues, but they don't have quite the same ability to move on their own or last as long as these monocytes do. And the monocytes are destroyers of anything that they can get a hold of and swallow. That's bacteria for sure, infected or diseased cells from your own body, foreign cells, foreign tissues, anything that they can recognize as foreign or malfunctioning or alien, they will go ahead and attempt to eat. The lymphocytes fight off infections by throwing out antibodies all over the place. So anyway, a little rundown of your white blood cells right here. In this particular case, organized by internal organization rather than abundance. Neutrophils the most abundant, lymphocytes the next most abundant, monocytes the third most abundant, and then a pretty even split between these two, which are quite rare, the eosinophils and the basophils. And as it says right here, 1% of your total blood cell count is typically these white blood cells. So 99% of your blood cells are going to be the red blood cells. These guys have nuclei. They last longer. They can respond to their environment and all that kind of good stuff. For instance, lymphocytes, when they're stimulated, pump out large amounts of antibody proteins. Well, protein production requires transcription and translation, so they've got to have DNA in their nucleus to transcribe to produce those antibodies. A little bit about the function of platelets. And what platelets do is if they make contact with exposed collagen, which is an extracellular structural protein in animal tissues, you don't have collagen normally anywhere exposed inside the circulatory system. This is a collagen-free zone right here. Collagen is a trigger for platelets to become sticky and to swell up, and then to release signal molecules that stimulate other platelets to swell up and become sticky, and also to stimulate the clotting function, the clotting response of your blood. And so there's that clot forming. As the clotting proteins become active, they precipitate out as a solid mesh, and the platelets stick to them. Eventually, red and white blood cells get all tangled up in this mess as well, and all of that collectively creates a clot right here. One other big function of the platelets is they release growth hormone stimulants that stimulate fiber cells of the connective tissue boundary. The outer boundary of this capillary is defined by connective tissue. The fibroblasts of that connective tissue will start reproducing really quickly to create scar tissue right here, fibrous scar tissue. And the endothelial cells, the lining cells right here, wherever they're missing, those cells will be triggered to divide as well, to regenerate the endothelial lining of the inside of the capillary bed here. So all of these cells are endothelium. It's a specialized version of epithelium. So a little bit about vessels and veins. So vessels is a collective term for tubes that carry blood. And those can be divided up into arteries, which are very large, muscular, high-pressure tubes leading away from the heart. Leading away from the heart. That's the definition of an artery, taking high-pressure blood away from the heart, all right, towards a capillary bed. Arterioles are the smallest of those arteries because there's one big artery leaving the left ventricle, one big artery leading the right ventricle. Those things branch and branch and branch and branch again as they distribute out into various organs of the body. And the finest of those branches is called an arteriole. And that arteriole opens up into a capillary web. And then those capillaries collect back together in a very small vein called a venule, just like an arteriole, venule, same idea, or venules. And those venules collect together into larger and larger vessels, the veins. And those veins collect together until finally we have two big veins leading back to our heart, the superior vena cava and the inferior vena cava, the superior coming from the upper part of the body, the inferior coming from the lower part of the body. And those take low pressure blood back to the heart. All of your arteries and veins are built, as it says right here, of 4 tissue layers. These are referred to as the tunics. So a tunic is kind of an term for like a shirt that you slip over yourself, but it also means an outer covering of anything if you go back to its Latin roots. So think of tunic, hopefully, you've got a definition now for a tunic if you're familiar with the piece of clothing called a tunic. Anyway, there's the tunica externa, the tunica media, which either is muscular or fibrous. That's 2 layers right there. And then the tunica intima, which is the endothelium on the inside right here. So there's 4 tissue layers, 3, significant tunics right here, one of which has 2 components. All right. So there's an elastic connective tissue outer covering, the tunica externa. And then we have tunica media, which is adjustable, and that's where the smooth muscle comes in right here. And it also is composed of quite a bit of connective tissue. And the ratio of these 2 varies quite a lot depending where you are in the circulatory system. And then inevitably there's a tunica intima, the endothelium in the center right here, facing the fluid. Additionally, when we look at veins, there's one other feature of veins that's not present in arteries, and that's one-way valve. So mechanically, this is a one-way valve. If you increase pressure on the fluid in this side right here, that pushes the flaps of the valve open, and fluid goes in this direction. If pressure here becomes higher than pressure right here for whatever reason, that pushes the flaps of the valve together, and they close. And so that means that this thing preferentially only allows blood to move in this picture from the bottom to the top. It's a one-way structure. Because of the very low fluid pressure in the venous system, we have to have valves to ensure unidirectional movement of blood. Now, in vertebrates, there is another component of the circulatory system that you don't normally think of. You may be aware of lymph nodes in the lymphatic system. What you may not be aware of is that it's really a 3rd branch. Arteries, veins, and lymphatic veins, the 3rd branch of the circulatory system. And what this does is, this is kind of equivalent to just the venous system. So imagine just capillaries joining into small veins, venules, joining into bigger veins, and finally one big vein leading back to the heart. That's really what the lymphatic system looks like as well. They've got these blind little capillaries that start in tissues of the body, and those capillaries join together to make tiny lymphatic veins, and those lymphatic veins join together finally to make 2 great big veins called the subclavian lymphatic veins, or lymphatic ducts, that join into the venous system right before the heart. So they dump into the blood supply right before the heart. And what these do is suck up leaked out fluid from your tissues, and that is the plasma that leaks out of your blood. Some of that's sucked back up by your blood, but some of that fluid pools in tissues, and it's sucked up by the lymphatic system and pulled back towards circulation. So this is a way of sponging up liquid in your tissues of your body and returning that liquid to circulation via the veins right here. And this also allows for an immune function. Where capillaries join together, where lymphatic capillaries join together, we have lymph nodes. And those lymph nodes are replete with monocytes, the inert version of an amoebocyte, and full of lymphocytes, T and B lymphocytes, which produce antibodies when they're triggered by the presence of an antigen somewhere nearby. And so the lymphatic system also is a sensory system in that those cells sitting in those lymph nodes are chemically tasting the liquid sucked up from the tissues of the body, and if they detect signals that indicate viral or cancerous or bacterial or parasitic activity in that tissue that's passing through that lymph node, those white blood cells will mobilize. The amoebocytes will actually march back downstream. They'll go back to those capillaries and leave those capillaries and go into the tissue looking for what they can identify. And in the meantime, the lymphocytes right here, in place in those lymph nodes, will multiply, so they'll become much more abundant, and that's why the lymph node swells, and they'll start pumping out antibodies that are sucked up by the lymphatic fluid, circulate all the way back to the heart, and then are pumped out to the body, and when they, and therefore protect your entire body from the thing that's infecting some particular location. So that's part of the immune response, which is quite fascinating. So a little bit about the lymphatic system. All right, as far as anatomy of the heart, all I'm going to require you to know is the 4 chambers, where we have valves and why we have to have valves. So we've got a valve leading from the atrium to the ventricle. So this valve right here, it maintains blood in the ventricle and keeps blood from going back into the atrium. The atrium is a receiving chamber. It's a very weak pump. The ventricle is a pumping chamber, very, very powerful right here. And if there wasn't a valve right here, when the ventricle contracted, we just blow blood back into the veins, which would be of no use. So we want the blood to go in one direction. And then we've got a valve from the ventricle into the artery that it serves for the same purposes. We blow blood into the artery, but the artery is a high-pressure tube. So if we leave this gate open right here, the high pressure will cause the blood to squirt back into the heart. We don't want that, so we've got a one-way valve. So that ensures blood can fill the atria here without competition from blood coming out of the ventricles. When the ventricle relaxes, it actually sucks blood out of the atrium into the ventricle. When the ventricle contracts, that closes this valve right here. For the record, it's called the tricuspid valve. That's the name for this valve right here, because it has 3 lobes. And that means that blood can only go out through this valve right here, where we have this aortic valve right here, this pulmonary valve, because this is the pulmonary circuit right here. This artery goes off to the left lung and the right lung, and one-way valves. Same thing for the left side of circulation. Blood comes from the atrium, goes through what's called the bicuspid valve, which is a valve separating the atrium from the left ventricle. Under pressure, the left ventricle blows blood out through the aortic valve right here into the aorta, one great big artery which branches off to go to all parts of your body in systemic circulation. Know the 4 chambers, where each chamber directs blood to in turn. So the right atrium directs blood to the right ventricle. The right ventricle directs blood to the lungs via the pulmonary circuit, and so on and so forth. And know the 4 valves. That's it. Hopefully this is not too surprising, but the cross-sectional anatomy of the lung is very much like your arteries and veins. There's a tunica intima, which is an endothelial layer right here. Unfortunately, they've chosen to name it the endocardium. It's a specialized epithelium, endothelium, which we find in the circulatory system, separating blood from the muscle tissue behind it. There's going to be a little bit of connective tissue and some capillaries here to keep the endothelium itself alive. And then a great big layer of muscular tissue, that's the myocardium, and then a layer of connective tissue, rich in protein fibers and all of that sort of stuff, and that's the pericardium right here. And then we actually have a fluid-filled space, and there's a mesothelial layer facing this right here, epithelium facing this liquid right here. And then this is the sac that your heart is sitting in. So if you dig into the chest cavity, if you're careful, you'll find the heart is enclosed in a membranous bag called the pericardium, peri for around, and that's a fluid-filled sac, and there's a tiny distance between the pericardium and the heart right here, just in the same way that your lungs are suspended inside the pleural cavities, and there's a fluid-filled layer between the surface of your lung and the inside lining of your chest cavity. Same deal with the heart. And so we've got a tunica intima, tunica media, tunica externa right here, and then on top of that we've got this great big bag that the heart is sitting in. I've talked about the valves, I've talked about the chambers, so that's really covered. We don't need to go over that anymore in this, but we've got some reminder material right here. Just details, I do need you to know the 4 valves. So the atrioventricular valves are simply the valves that separate an atrium from a ventricle. There are 2 of them. One is called the tricuspid, that's on the right side. There's one called the bicuspid, or unfortunately yet another name, mitral valve, on the left side. And then these so-called semilunar valves, which are the valves that lead from either ventricle out to an artery. There's the pulmonary one, and there's the aortic one. All right. All right, a little bit about the mechanisms for heart contractions, and that'll be the roughly the end of this presentation. And so this applies to all hearts, hearts in Daphnia magna, hearts in ants, hearts in anything. The basic ideas hold the same. The details will be different, of course, but the basic idea is the same. So muscular tissue consists of a wave of electrical activity across muscle cells. Basically, the polarity across the cell membrane changes from positive on the outside to negative on the outside rapidly, and that process of electrical depolarization spreads across muscle tissue as it's contracting. Since there's electrical activity, we can measure that electricity, the change in voltage, and if we graph it, this is positive and this is negative in this direction right here, 0 being somewhere right about here. This will be the change in voltage over muscle tissue that you would pick up, when they do an ECG, an electrocardiogram. This is the wave that they get right here, measuring collectively the change in voltage across the heart. And this is the pattern that you're looking for right here, and it kind of tells a story if you know how to detail it right here. So as you can see, this first little hump right here is in purple. That means that's what they're talking about in the picture up above. This is, the change in voltage, which means some cells are in the act of going through the contraction phase. And then that moves to another location right here. There's another action going on right here, etc., etc. So the way it works is, and we're showing in purple right here, what tissue is involved in this process. You've got some cells called pacemaker cells right here, which is a characteristic of smooth and cardiac muscle tissue, where some of the cells are capable of stimulating their own contraction. They kind of go off like a timer. And that's what the pacemaker cells here in the sinoatrial node are. That's the pacemaker. When somebody has a medical pacemaker put in, what happens is medically this thing is knocked out, so it doesn't control your heart anymore, and then an electric probe is put in this position right here, or not a probe, but an electrical fiber, and the pacemaker sends a little jolt of electricity that starts the tissue around here to contracting. One cell stimulates its neighbor. That's the way cardiac muscle tissue works. When one cardiac muscle cell contracts, its neighbor starts contracting, and that triggers the neighbor to contract, and so on and so forth. So from this node right here, we spread a wave of contraction across both atria. And so the two atria contract very weakly and push blood into the relaxed ventricles. As that wave hits this thing called the atrioventricular node, which is another little nervous structure in the heart right here, that's basically the receivers, the dendrites of a bunch of neurons, and those neurons fire off a signal down their axons leading to the bottom of the ventricles and the wall separating the two ventricles, and ultimately all the way through the musculature of the ventricles. So as the atria are contracting, this internal nervous system that's just of the heart, here's kind of the brain part of it, here are the axons sticking out, triggers the ventricles, causing them to depolarize, and that's what's going on right here for just a minute. And then the ventricles contract very, very powerfully, and that's what this big spike right here is as we go through that ventricular contraction. And that is one cycle of contraction through the heart. So if you're actually going to look at this, and we'll go ahead and do that. All right, we're looking at a beating heart animation right here. That's not too bad. You can see the valves in operation. All right, so that animation showed you in an animated sense these steps right here of a heartbeat at work. A couple of last little bits right here. So some terms. You've heard of blood pressure, and you know that your blood pressure is usually one number over a number, something like 120 over 60. So the 120 would be the systole or systole. That's the highest pressure right after your ventricles have contracted, the highest pressure in your arteries. That's systolic pressure. And then diastole is the pressure in your arteries when the ventricles are relaxed and the arteries are, blood is squeezing out of the arteries into the capillaries that they serve. And so we get that systolic and diastolic difference in blood pressures. The lub dub, the heartbeat that was included in that animation, That's the sound of the two pairs of arteries opening, or not opening, but closing. They don't make any noise opening, but when the two halves slap shut, or the three halves in the case of the tricuspid valve, there's an actual sound that can be heard. So that's the valves closing. The atrioventricular valves are the lub, the first bit, when the ventricles start contracting. The AV valves close so that blood doesn't go backwards into the atria. And then the dub is the closing of the semilunar valves after the ventricles relax. So just as the ventricles relax after compressing, and blood tries to flow back from the arteries back into the ventricles, that's when the semilunar valves leading to the arteries coming out of the heart close, and that's the dub of the dub 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 dub, usually written down as lub dub lub dub lub dub. All right, so once again, excitable tissues in the heart consist of two particular locations: the sinoatrial node, which is the pacemaker, and then cardiac muscle as it's contracting. When cardiac muscle adjacent to the atrioventricular node, when this muscle tissue here contracts, it causes an electrical depolarization of the AV node, and that's more of a nervous structure. And then that transmits a signal to all the rest of the fibers of the heart's intrinsic nervous system. So that is, it's not really a brain, but it is a nervous system that's strictly in the heart and of the heart right here. And from your brain, you do have nerves coming down, making contact with the sinoatrial node right here, and desensitizing it, which slows the heart down, or exciting it, hyperstimulating it, which speeds the heart rate up. And so that's how your brain and your endocrine system and various other things control your heart rate. It's by controlling primarily this little bit of tissue right here, the sinoatrial node. And the autonomic nervous system is the part of your nervous system that controls heart rate. If you cut those nerve fibers from your brain to your heart will carry on beating at its natural intrinsic rate, which is just based on this thing doing its own thing, and that's about 75 beats a minute, roughly. A couple of schematics that show some important relationships between different characteristics as we go through flow of the blood through the circulatory system. So here we're exiting the heart, going all the way down to capillary beds, and then we're returning to the heart, and this is systemic circulation. The graphs would be similar, the values would be a little different for pulmonary circulation. And so one thing, vessel diameter. The arteries coming out of the heart are very large in diameter, and as we get closer and closer to the capillaries, they get smaller and smaller and smaller in diameter until the capillaries themselves are very, very tiny, just slightly wider than one red blood cell, so on the order of 200 to 300 nanometers across as a size, really quite small. And then the vessels returning to the heart get larger and larger. Note that the veins, the vena cava, these are the great big veins leading back to the heart from the upper body and the lower body respectively, are much larger. The inferior vena cava anyway is much larger in diameter than the aorta leading off to your heart right here. Immediately what that's going to cause is blood will have to move faster through this smaller tube. Since the volume is relatively fixed, blood is going to move slower through this bigger tube, since the same amount of blood per unit of time is moving through all of this stuff. So it's going to be going faster here, much slower there. Okay. B, total cross-sectional area of the vessel. So if we look at the aorta and just measure its area, if you cut it across and measure the area of the opening into the aorta, it's some small fraction of 1,000 centimeters squared right here. It's it's a pretty big vein, but or vessel, I should say. Anyway, as we reach towards the arterioles and then most prominently the capillaries right here, we see that the cross-sectional area of all the capillaries added together is quite a bit more than the cross-sectional of just the aorta that feeds these capillaries. And then the same thing, the capillaries join together in venules, small veins, big veins, larger veins, the vena cava. And as I said before right here, the vena cava has a larger cross-sectional area than the aorta, and that's represented here. The vena cava is still a bit higher up off of the zero line here than the aorta, but the capillaries are a much bigger cross-sectional area. So what that means is, as blood flows down those arteries, two things drop consistently as the cross-sectional area grows and grows and grows, and that is the pressure of blood. It falls and falls and falls. It starts at, say, an average of 100 millimeters of mercury right here. As it passes through the capillary beds, the standard values are 40 going in, 20 coming out millimeters of mercury, and dang close to zero by the time we get to the vena cava going back to the heart. So it's almost like the heart sucks blood back into it because the pressure is so low. And the speed at which blood flows. Small tube, blood must move quite quickly. Big tube, or in this case, millions of tiny tubes added together to make virtually one great big tube, blood flows very slowly, and then speeds up again as the diameter of the tube gets smaller and smaller. The blood speeds up again, so the blood flow, the rate right here. And this is good. We want to get blood from the heart to capillary quickly, hence a lot of the distance is very high speed. In the capillaries, we want the blood to move slowly, so there's lots of time for exchanges between the tissues right here. Gases, nutrients, hormones, water, salts, everything. And then we want the blood to move pretty quickly back to the heart. So as that total cross-sectional area comes back down again, necessarily the velocity goes back up. But we don't gain any pressure right here. Right, and so yeah, let's talk about this. Systole versus diastole. Again, that's the difference between high blood pressure and low blood pressure right here. Ventricular contraction, ventricular relaxation. Those typical values measured at your arm, your upper arm, are going to be 120 over 60 as sort of benchmark values. If we go from your arm further down to an artery leading, closer to tissues right here, recognize where's the pressure right here. Yeah, the pressure must continuously go down. The average pressure drops from 100 to 80 to 60 to 40, when we actually get into capillaries. So if you measure your blood pressure, further away from your heart, that pressure is going to be lower. So we try and measure it at a standard location, which is the upper arm on the left side, which is closer to the exit from the heart. And in reality, the pressures inside the heart are somewhat higher. It's more than 120 over 60. It's going to be like 140 over 80 or something like that. And then people with hypertension are going to have elevated values anyway. The high blood pressure is a result of clogging along the tubes right here. So to get the same pressure in the capillaries and the same flow through the capillaries, if you have clogging in these arteries, you're going to have to raise the pressure in the beginning of the system in order to get the same thing occurring down here. There's a feedback mechanism that ensures that the blood flow through your capillaries is a certain minimum, and what happens is we increase the rate of contractions of the heart and the ferocity of the contractions of the heart as well. And ferocity is a kind of strong word, but the intensity of the contractions, and we also contract smooth muscle in the arteries to raise blood pressure. We'll also contract smooth muscle in the veins right here so that blood piles up backwards right here, and it increases the overall pressure so that we get the correct pressures at the capillaries. And that's the basis for hypertension. This next image right here is the mechanics, previously was the physiology for the hypertension situation. Here's what causes that adjustment in pressure. Blockages in arteries right here. This is arteriosclerosis, and that's where a lipid-rich and protein sort of mixture of materials creates this plug of stuff between the endothelium and the tunica media right here. So that's the endothelium right there. There's the tunica media behind it, smooth muscle and or connective tissue. And then we got this block of stuff called plaque right here. And the result is it narrows the opening of the artery right here. So we have to increase pressure to get the same amount of blood per unit of time through this decreased little opening right here. And that's the accumulation of coronary disease and arterial disease, that high blood pressure is a symptom of. Two terms right here. Atherosclerosis is the accumulation of fatty material right there. Arteriosclerosis, when it's a hardening, due to some mineral depositions like calcium right here. All right. We talked about these three hormones in the endocrine system, three of these. We didn't talk about A and H right here, so I'll talk about that one right here. So three hormones that contribute to blood volume and therefore blood pressure regulation. Antidiuretic hormone changes the rate at which blood is filtered by the kidneys. That's diuresis right here. So antidiuretic hormone causes less liquid to be filtered out of the blood. And the action of ADH is to increase the permeability of the tubes in the kidneys to water by causing those cells to produce more aquaporins. And so under the influence of antidiuretic hormone, your kidneys reabsorb more water out of the filtrate, the stuff they've filtered out of blood that is the basis of your urine. Aldosterone, in concert with antidiuretic hormone, encourages or increases the rate of movement of water. Aldosterone specifically induces cells of the kidney tubules of the nephridia to reabsorb sodium. And if we reabsorb sodium, as you know, water follows by osmosis. Osmosis is made easier by the action of antidiuretic hormone, which induces the cells in the kidney tubules to produce a whole bunch of aquaporins, making the cells more permeable to water so that the salt can drag more water behind it via osmosis. And then those aquaporins are removed unless there's more antidiuretic hormone released, which causes the cells to put out more aquaporins. So it's a continual balancing act. Atrial neuritic hormone, neuritic or neuropeptic hormone right here, is released by the atria. If pressure is too high, if the walls of the heart atria, particularly the right atrium, are stretched too much, then that stimulates the release of this hormone, which is an antagonist to aldosterone right here. So it increases the dumping of sodium into the urine. Water therefore follows the sodium into the urine, or stays in the urine, and we reduce blood volume and therefore blood pressure. Then finally, I talked about nitrous oxide in the endocrine system lecture. Nitrous oxide is a gas. This is the nitrous oxide that, it's gas, NO right here. And it is produced locally by endothelial cells and other cells in the circulatory system and stimulates smooth muscle to relax and dilate. If the smooth muscle relaxes, blood pressure is going to naturally stretch those tubes open. The tubes will open to their widest diameter, if the smooth muscle is relaxed, the widest diameter they possibly can. So smooth muscle is always contracting to some degree, but the level of contraction of that smooth muscle is a function of nitrous oxide, it's a function of atrial neuritic hormone, and it's an effect of other hormone stimulants that are released into the blood. All right, and that is everything that we need to talk about in the circulatory chapter right here.